Hello everyone, welcome to my YouTube channel. Today I come up with this incredible gown. How do you find it? Do you like it? If you do, please show me in my video by liking it. Again, don't forget to subscribe to my YouTube channel. Thank you so much for you subscribing. Today I am going to discuss about cellular base of life. Organisms are made of one, that is single-celled or unicellular or more multicellular organisms, which need a supply of energy and molecules to carry out life processes. The cellular base of life is one of 15 big ideas of science in the base evidence science teaching collection. Therefore, cellular base of life is one of 15 big ideas. The cell theory, the unified cell theory states that all living things are composed of one or more cells. One-celled organisms, unicellular, there are unicellular organisms, or more cells that are multicellular organisms. So organisms are made of at least one cell, that is unicellular, or many cells or multicellular. There is no organisms which do not have cells. The cell is the basic unit. When we say the basic unit, structurally and functionally. And it is also called the smallest unit of life. Why it is the smallest unit? As there are smaller things such as atoms below the cells. They are called the smallest unit because cells are the smallest things that can live independently. One reason. The other reason why cells are called the smallest unit is the cell is the smallest unit of life that can divide, multiply, grow, and respond to stimuli from the environment. The things which below the cells, which are smaller than the cells, do not do these things. Therefore, that is why cells are called the smallest unit of life. The cell theorists are Sheldon and Schwann. This is just to acknowledge these scientists. Biochemical activities of cells are dictated by their shapes or forms. Dictated means controlled or directed, and by the relative number of their specific subcellular structures. What are the specific subcellular are? Just we are going to discuss here. They are the organelles. This is an animal cell which contains different organelles. Just you can see here different organelles. For example, this is a nucleus which is composed of nuclear pores. The pores at the surface of the nucleus, then DNA. This is a thread-like structure. This is a DNA. Then nuclear envelope, the outer cover. Envelope means just like a post. So the outer cover. Then nucleolus. Nucleolus is different from nucleus. Okay, nucleolus. This region is nucleolus. The other organelle in a cell is cytoskeleton. Okay, the thread-like structure. We will see each of them. Then rough endoplasmic reticulum, here rough one, and then smooth endoplasmic reticulum, this one. Then free ribosomes, the ribosomes. When we say free ribosomes, the ribosomes which found without attaching to this endoplasmic reticulum. Then cytosol, the fluid part of the cell. Yes, it looks like water. The color looks like water. Is cytosol, or we call it also cytoplasm. And lysosomes, just here you can see a lysosome which contains different molecules within it, like a container. Lysosomes are like a container. So these are some of the organelles. Just we will discuss each of them here in this video. So cell organelles. An organelle is a specialized subunit within a cell. It is a subunit within a cell that has a specific function. Each organelle has a specific function, their own function. In eukaryotes, an organelle is a membrane bound. It has a membrane, okay? A membrane bound structure found within a cell. Just like cells have membranes to hold everything in, okay? Cells, just we will see these things, the membranes. Cells contain membranes, okay? Cells are enclosed by membrane. So like that, these mini organs that are organelles are also bound in a double layer of phospholipids. Phospholipids are the major components from which the cell membranes are made of. This is to insulate their little compartments. Okay, there means here the organelles. The organelles little compartments within the larger cells. Compartments means just like a room just where you are sitting now. Okay, compartments. Prokaryotes are cells that do not have membrane-bound organelles. Okay, the organelles which found in prokaryotes are not membranous or do not contain membrane. Organelles based on membrane covering can be divided into two, membranous and non-membranous. From the name, just simply, membranous means having membrane, and non-membranous means do not contain membranes. Membranous organelles include endoplasmic reticulum. There are two kinds of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. We will see them. Golgi bodies, mitochondria, chloroplasts, nucleus, lysosomes, peroxisomes, and vacuoles. These are membranous organelles. Non-membranous are ribosomes, both 70S and 80S. 70S ribosomes are in prokaryotes and 80S are in eukaryotes. What is S? S indicates here svedbergs. Svedbergs, a unit used to measure how fast molecules move in a centrifuge. This is a centrifuge. This is a tool or apparatus. So this is related with the centrifuge, the svedbergs. A svedberg unit or svedberg, we can use S or SV, which is non-asymmetric unit for sedimentation coefficients. Sedimentation. When things are centrifuged here in this machine, then it will form a sediment. Then let's see each of the organelles. The first one is the nucleus. Nucleuses are oval or spherical shaped. Oval means like egg, spherical means like sphere. Sphere means like the spherical shape of globe. These are largest central structure in cells. They are largest in a cell and they are central, surrounded by a double layered membrane. Okay? Nucleus are double layered, having two layers. Governs most cellular activities. Governs means controls. It controls most cellular activities and serves as the cell's master. So nucleus are called cell's master because they monitor protein synthesis activity. So proteins are very important in every function of cells. Therefore, these important molecules synthesized by the control of the nucleus. For example, DNA directs protein synthesis because the genes which will be converted into proteins are within DNA. Okay, so DNA codes. Therefore, DNA directs protein synthesis and serves as a genetic blueprint during cell replication. DNA gives codes or instruction for directing synthesis of specific structure and enzymes or proteins. Enzymes are proteins within the cell. There are three types of RNA are involved in the protein synthesis process. Okay, in this process there are three RNAs: messenger RNA, abbreviated as small m, capital RNA, mRNA, ribosomal RNA, small r, RNA, small t, RNA, transfer RNA. We will see these things in another video. Generally, the nucleus may be rounded, rounded. For example, in hepatocytes, hepatocytes means liver cells. They are rounded in liver cells. Indented means just like zigzag, okay, just like zigzag. So they are indented or segmented, for example, in neutrophils. Neutrophils are a type of white blood cell. Binucleated. Bi means two. Nucleated refers to for nucleus. So having two nucleus. There are two nucleuses in some cells. For example, in parietal cells and cardiac muscle cells. Cardiac related with hearts. So heart cells contain two nucleuses. Multinucleated. Having many nuclei. Multi means many. So having multinucleuses. For example, in osteoclasts, these are a kind of cells. Skeletal muscle cells, which contains many nucleuses, very large nucleuses, because of having many DNA. For example, in megakaryocytes. What is megakaryocytes? Mega means large, so large nucleus cells. Karyo indicates nucleus, so large nucleus cell is a large bone marrow cells with a lobated nucleus responsible for the production of blood thrombocytes or platelets, which are necessary for normal blood clotting. Nucleuses may be absent in some cells, for example, in mature erythrocytes. Erythrocytes means red blood cells. And also in blood platelets, there is no nucleus in these cells. When you say mature erythrocytes or mature red blood cells, after the process of making of the red blood cells, after some hours, the cells will mature. The newly synthesized red blood cells contain nucleus, but after maturation, after some times of its synthesis, it will lose its nucleus. So the mature red blood cells do not contain a nucleus. The nucleus is surrounded by a nuclear envelope. Envelope means the outer cover, and contains chromatin and one or more nuclei. For example, the nuclear envelope, this is cover like post. Post means just the envelope in which you will put your letter when you want to send to your friend or your family. So like a post which surrounds nuclear material, the material which found in the nucleus. It covers the materials which found within it, okay, within the nucleus. Nuclear envelope consists of outer and inner membrane. Okay, this has two membranes, the outer membrane and the inner membrane. Perforated, which means pierced at intervals by nuclear pores. These are the pores on the surface of the nucleus. Through these pores, most ions and water-soluble molecules transfer between the nucleus and cytoplasm. Chromatin, this is one of the components of a nucleus, which means chromatin means colored material. This naming is because of it easily stained for viewing with microscope. Okay, what is staining means? Staining means a color or a material or object by applying a penetrative dye or chemical. Dye means color. To enhance what? Visualization. Why we stain it? Just to enhance visualization. The contents of most cells here, add T here. Okay, the contents of most cells, which are 70% water by weight, to impede the passage of light rays. Therefore, when the molecules are watery, as you know, water are transparent. Therefore, transparent things cannot be seen easily. Okay, water is colorless. Therefore, it is difficult to observe through the microscope. So, what will be the solution? Adding colors, okay, to enhance what? Visualization. That is it. The other component of nucleus is nucleoli. It says nuclei, the plural form of what? Nucleus. Nucleus is singular, nuclei is plural. So the nuclei of most cells contain one or more lightly stained structures that we call nucleoli. Okay, nuclei is different from nucleoli. Okay? Nuclei is the plural form of nucleus, and nucleoli is the lightly stained section of the nucleus. So this is called the nucleoli that actively engage in synthesizing of ribosomes. Okay, ribosomes synthesis takes place in this structure. This is a nucleus, and the lightly stained part whitish. Therefore, we call this nucleoli. The cytoplasm or cytosol. We can say cytoplasm or cytosol. This is the other organ, the second organ in our discussion. The first one is nucleus. This one is the second. This is a complex jelly-like marrow. Jelly means the sticky-like compound. This occupies about half of the total cell volume. Okay, this is a large amount. It covers a large amount of a cell, half of the cell. The other organelle is Endoplasmic Reticulum, abbreviated as ER. This is a fluid filled, which is filled with what? Fluid. The fluid-filled membrane system extensively present throughout the cytosol. It occupies a large space extensively. There are two different types of ER, smooth and rough ER. The smooth ER is a mesh work of interconnected tubules, whereas the rough ER projects outwards from the reticulum as stacks of flattened sacs. Okay, we will see. The smooth one is look like tubules, or tubules means tube-like structure, and the rough one is sacs. You know, sac. Sac means the container in which you can put teff, maize, and other stuffs. So the rough one looks like the sacs. Though different in structure and function, they are continuous with each other. Both smooth ER and rough ER are different in structure. They are different in structure and their job or their function, but they are found together continuously or together. Rough one and the smooth one. The rough endoplasmic reticulum, the outer surface of the rough ER contains dark particles called ribosomes, which make the surface looks rough. That is why it is called rough, because for our eyes it looks like what? Rough, because it contains ribosomes, which are ribosomal RNA protein complexes that produce protein under the direction of nuclear DNA. Smooth endoplasmic reticulum we call smooth ER, since it does not have ribosomes. It do not contain ribosome. So for our eyes it looks smooth and does not produce proteins because it do not contain ribosomes. Ribosome is the stage or the place where proteins can be synthesized. Protein synthesis takes place in the ribosomes. As smooth ER do not contain ribosomes, there is no protein synthesis there. And smooth ER serves in packaging and discharging site for protein molecules that are to be transported from the ER. When we say ER, here rough ER, okay, rough ER. because protein synthesis takes place in the RAF ER. Because the RAF1 contains what? The ribosomes. Therefore, the protein synthesis takes place there. When you say discharging, just distributing or deploying. Okay, the deploying or distributing of the proteins from where it is synthesized to where it is needed in cells. Portions of the smooth ER for transport vessels. When we say vessels, just a vessel is a container like your bag, for example. Therefore, portions of the smooth ER form transport vessels which contain the new molecule rapidly. What is a new molecule? Within the bag or within the container is the protein. Okay, the proteins are within or rapid in the vessels. Transport vessels, the vessels which we have seen. So transport vessels move to the Golgi complex. This is another kind of organelle which we will discuss just later on. So the transport vessels move to the Golgi complex for further processing of their cargo. Here the cargo indicates what? The proteins. After processing in the smooth ER, the protein is made in the RAF ER, then processed or modified in the smooth ER, then again for further processing it will be transported to Golgi complex. In this case, cargo means proteins. What is cargo means? Cargo is a bulk goods. Okay, goods. Goods are just clothes, just you are wearing, or other things you use that are goods. So bulk goods conveyed by water. The transportation system may be on water bodies or through air by airplane or on land by car or like that. Therefore, that is cargo. Smooth ER has additional functions. Why we say additional? Because its main function is what? Packaging and discharging. Just we have seen in the previous slide. Okay, in this case, the primary function of the smooth ER is packaging and discharging. In addition to that, it also does these activities. For example, it synthesizes lipids. The smooth ER synthesizes lipids. Making of lipids. Synthesize means making. But the smooth ER do not synthesize or make proteins. Okay, they do not make proteins. They can make what? Lipids. They do not make or cannot make proteins because they do not have what? Ribosomes. Another function is detoxifying harmful substances. The smooth ER detoxify, just toxic. So detoxifying means converting the toxic things into non-toxic. Therefore, it detoxifies. It converts toxic things into non-toxic and plays a crucial role in the process of muscle contraction. These are additional functions of smooth ER. Just structurally, the smooth ER, just by picking from this large cell component and magnifying, the endoplasmic reticulum generally looks like this. Okay, this is RAF1. Okay, RAF endoplasmic reticulum because it contains what? Ribosomes. So for our eyes, it is what? RAF. Okay, this dot looks like RAF. Okay, this is a large cell from which also the ER is enlarged. So this is just the RAF1 is like sacs. Okay, like sacs. And this one is like tube. Okay, the structure is like tube and it do not contain ribosomes, so it looks like what? Smooth. Okay, when you say rough and smooth, just for example, you can compare your skin. When you touch your skin, it is what? Smooth. Touch the wall or touch some other mat or table, it is what? Rough. That is for touching. Just this is not for touching. We can't touch the organelle just for our eyes. Okay, looks rough and this one looks smooth. Golgi bodies, another kind of organelle is Golgi bodies. Multiple names. There are multiple names for Golgi bodies. It is also called Golgi apparatus, Golgi complex. Why it is called Golgi apparatus? You know, apparatus means what? Material or tools. Why it is called apparatus? Because, for example, other organelles such as lysosomes and peroxisomes can be made here in this Golgi body. So they are called apparatus. It is named after its discoverer, scientist Camillo Golgi. This is Camillo Golgi. The Golgi complex is associated with the ER, Endoplasmic Reticulum. The Golgi complex found together with Endoplasmic Reticulum and contains cells of flattened means flat, okay, flattened, curved membrane, enclosed, which is enclosed sacs. Okay, I told you sacs means the container in which you can put your tape, maize, wheat, and the like, which we call it cistrinum. This enclosed sacs is called cistrinum. The majority of newly formed molecules from the smooth ER. What kind of molecules can be made in smooth ER? It is what? Lipids. Okay, lipids. Okay, because additional function of smooth ER here we have seen is just making lipids. So these lipids from the smooth ER enter Golgi complex stacks. Okay, for what? For further processing or modification. Golgi complex performs the following important functions. These two important functions. One is processing the raw material into finished products. Raw means just before converting into the usable form. So this raw one will be converted into finished products. The product which can be utilized. There is no further processing if the product is finished. So in this case, raw indicates the protein from the ER are modified into their final state. Okay, the finalization of the protein modification takes place in the Golgi complex. So when you say raw material, the proteins, okay, the proteins will enter into Golgi complex and then finalized. The other function of Golgi body is sorting and directing finished product, that is proteins, to their final destination. Because where the protein synthesis takes place is in the ribosome, different cells require proteins. Then they ask the ribosome to make proteins. Then after making the protein in ribosome, then it enter into smooth ER for modification. Then again it will enter into Golgi complex for more modification. And then finally move into where it is required. So these are the two important functions of Golgi complex. This is the structure of Golgi complex. Okay, it is look like a sac in which the protein molecules can enter. Okay, you can look at the structure which is look like what? A sac. Okay, this is the same structure. Golgi body, this is the Golgi body. I brought to you just to clarify the structure. Okay, you can use this one or you can use this one. The other kind of organelle is lysosomes. Okay, lysosomes. These serve as the intracellular digestive system. Okay, it is not a kind of digestive system. You know the digestive system in our body, which starts from mouth, then esophagus, then intestines, small intestine and large intestines. But this is an organelle, not a kind of digestive system, but its function is like a digestive system. What is the function of digestive system? Just breaking down of the foods we eat and make ATP, breaking down into smaller components and making ATP. Therefore, the function of lysosome is what? The same. The name is from lys. Okay, lys means breaking or splitting. Some or soma means body. So lysosome means the splitting body, which breaks down or splits down the larger molecules. Lysosomes contain powerful enzymes, for example, such as glycosidases, proteases, and sulfatases. Glycosidases are the enzymes which act upon carbohydrates, proteases which act on proteins, and sulfatases on sulfur. These contain powerful enzymes capable of digesting and removing unwanted cellular debris. That is, just it breaks down unwanted wastes. Debris means wastes. The things which do not required by cells, that are wastes. So it breaks down these wastes and then remove from cells. For example, bacteria. You know, this is very interesting function because bacteria are germs, okay, which can cause disease. So this lysosome can break down bacteria, which are our enemy, to smaller components and then remove from our cells. Lysosomes can take up old organelles which are old and unwanted by the cell, such as mitochondria, and then break down into the component molecules. This process we call autophagy. Okay, this breaking down of the unwanted organelle or old organelles and removing process we call autophagy. This is a lysosome in which unwanted debris can enter and then be break down. This is a lysosome. From a large cell, when you enlarge this component, then you will see this one. This is a lysosome. The other kind of organelle is peroxisome. Peroxisome is a membrane enclosed sac. It is also a sac like lysosome, which containing oxidative enzymes and catalase that detoxify various wastes. It also contains catalase and antioxidant enzymes. This kind of oxidative enzymes, catalase, antioxidant enzyme, it contains, which decomposing hydrogen peroxide into harmless water and oxygen. The name peroxisome comes from what? Due to their role in peroxide metabolism. Peroxide is any of a class of chemical compounds in which two oxygen atoms are linked together by a single covalent bond. For example, in this case, these two oxygen, this oxygen and this oxygen are bonding each other. That is peroxide group or peroxide bond. So hydrogen peroxide, the molecule is H2O2. It is shorter and smoother than lysosome. When you look the function of the peroxisome, it is the same with lysosome. What is their difference? Their difference is this peroxisome is shorter and smoother than lysosome, and several hundreds may present in one cell. The other kind of organelle is mitochondria. It is called powerhouses of a cell because a lot of ATP is made here in the mitochondria. From our other discussions, you will understand this lot of ATP synthesis. For example, TCA cycle and electron transport chain takes place in the mitochondria, from which a lot of ATPs can be made. It is rod or oval shaped about the size of bacteria. Okay, this is because the size of the mitochondria is like bacterium size. What is the difference between bacterium and bacteria? Bacterium is a singular form. If it is one, we call bacterium. So the size is similar with bacterium. The mitochondrial shape and size looks like bacterium because MT here, mitochondria, is believed that it came from bacteria. Okay, it came from bacteria. We call this endosymbiosis theory. For example, the shape of the mitochondria is like this. If you look at most of the bacteria's shape, it is the same. Therefore, this is just the outer membrane of the mitochondria. This one is the inner membrane, and this one is the matrix in which TCA cycle and electron transport can take place. The space between this membrane, the outer membrane and the inner membrane, we call this space intermembrane space. Mitochondria, each is enclosed by a double membrane. Here is a double membrane, a smooth outer that surrounds the mitochondria, and an inner membrane that forms a series of infolding or shelves called cristae. Okay, the inner membrane folded like this, and the infolding, which folded to in, we call cristae, which project into an inner cavity filled with a jelly-like matrix. This is the internal space we call matrix, in which the ATP synthesis from TCA cycle and electron transport chain can take place. Therefore, from TCA cycle and electron transport chain, a lot of ATPs can be made. That is why the mitochondria is called powerhouse of the cell. Let's look at the general structure of the mitochondria. This is the mitochondria. For example, outer membrane, this is the outer membrane, then this one is intermembrane space. And the infolding is the cristae, and the matrix innermost space is the matrix, which you have seen in the previous slide. Mitochondria are unusual organelles in two ways. One is in the matrix, they have their own unique DNA called mitochondrial DNA. We can abbreviate this as MT for mitochondria. So mtDNA. It contains its own DNA. This is one unique feature of mitochondria. This is because of, as I said, it is believed that it came from bacteria because bacteria contain their own DNA. The ability to replicate themselves even when the cells to which they belong is not undergoing cell division. Cells divide always, you know that. So when cells divide, its components, such as organelles, divide. That is obvious and clear. But in an unusual way, the replication of the organelle mitochondria takes place without the replication of the cell which contain the mitochondria. These are the two unique features. One is it contains DNA, the other is it replicates without the replication of the cell which contains that mitochondria. The other kind of organelles are chloroplasts. Chloroplasts are useful in photosynthesis. The structure of the chloroplasts look like this. Here, the outer membrane, this is the outer cover. When you say membrane, always consider that it is the outer cover. So, outer membrane, then the inner membrane. It is double membranous, okay, having two membranes. You can easily see here. And this is a stroma, and here is thylakoid, the coin-like structure, okay, like coins. So these are like coins we call thylakoid, and this is a grana. Okay, the collection of the thylakoid is called grana. This is just the general feature of chloroplasts. I am planning to do about chloroplasts in the future video that related with the photosynthesis. So this is all about the chloroplasts for today. Then other kind of organelles are vesicles. Vesicles are membrane-bound sacs. They are sacs. Sacs means just as I told you repeatedly, like a container, okay, like your bag. Okay, so vesicles are membrane-bound sacs that are used to store or transport substances. What you will do with your bag? You can store your stuffs, yes, your things, or you can transport your things or your stuffs with your bag. So like that, vesicles is the same thing. Its function is like your bag. Vacuoles are just simply the larger vessels. If you understand what is vessels, so vacuoles are the larger vessels. These are just the structure of vessels, in which just consider that in this structure there will be what? The molecules which can be transported by the help of these vessels or vacuoles. The other kind of organelle is cytoskeleton. Cyto means cell, skeleton means just relate with human skeleton. You have learned it in the high school or your lower grades. So, the skeleton of the cell. Just consider that what happens if there is no skeleton of a person. Without skeleton. Skeleton means just bone part, the hard part of the body. What happens if there is no this bone and hard part of the body? Okay? It's difficult to think. Is that not? The body will become collapsed. Like that, cytoskeleton is the same function with the skeleton of human. Cytoskeleton is a complex protein. It is a protein, as proteins are very important in all or almost every kind of cellular function. It is here also cytoskeletons are proteins. This is a complex protein network that acts as a bone and muscle. Okay, just like your bone and muscle. Without your bone and your muscle, the other part is what? Water or fluid part. Then you don't have such kind of structure. Just consider your structure or your morphology or your shape. Without muscle and bone, you are just nothing. So cytoskeleton is like that. You like your bone and muscle. Cytoskeleton necessary intracellular scaffoldings. Okay, what is scaffolding is just we will see here. Necessary intracellular scaffoldings. Intra means within one cell. So intracellular scaffoldings. It is a necessary. It is must. Within a cell, the existence of cytoskeleton is must. So necessary intracellular scaffoldings supports and organizes cellular components, arrangements, and to control their movements. It controls also the cellular movements. This provides distinct shape, size to the cell. The cell shape and size is determined by cytoskeleton. Like your shape and size is determined by your bone and muscle. What is scaffolding means here? Scaffoldings are just consider the new building. These are the scaffoldings through which the workers can move up and down. That is what we call scaffolding. Cytoskeleton has at least 4 distinct elements. 1. Microtubules. Micro means small, tubules means tube-like structure. So microtubules, these are also proteins. Generally, cytoskeletons are proteins, therefore, these all are proteins. Microtubules, microfilaments, intermediate filaments, microtubular lattice. These are the 4 distinct elements of cytoskeleton. When we say filaments, the thread-like structures. Generally, cytoskeletons determine or provide the shape of cell, structural support, organizing its contents, the cellular contents, for substance movement through cell like cilia, flagella, and intracytoplasmic vessels, and it also contributes to movement of the cell as a whole. Okay, for the movement of substance through cell and also movement of cell as a whole. This is a cytoskeleton structure. Just this is outer cover of the cell, and this one is the outer cover, and this, for example, this is a microtubule. Okay, just the tube-like structure. This is very small tube. Just we are observing is very magnified by microscope. Let's consider very small. Then microfilaments, just thread-like structure. These are the thread-like structure, just which is used as scaffoldings, okay, to maintain this shape. Without this microtubule, this structure, the cell will come together and be collapsed. Okay, that is very important in maintaining the shape of the cell and supports the cell. The other kind of organelle is plasma membrane, or the other name is cell membrane. You can say plasma membrane or cell membrane. I am just using C.M here for cell membrane. Cell membrane is extremely thin layer of lipids and proteins. The main components of the membrane, cell membrane or plasma membrane, are lipids and proteins. What kind of lipids? The lipids which contain phosphates, so phospholipids, forming the outermost boundary of living cell and enclosing the intracellular fluid. So it is a boundary. So let's say this is one cell, cell 1. This is another cell, cell 2. Therefore, this cell is bounded by this membrane and the other cell is bounded by this membrane. Therefore, it used as the boundary, okay, to separate one cell from other cell or the other environment within the body. And it is selectively permeable. Okay, selectively means it chooses molecules based on their size. It is not totally impermeable. Impermeable means do not allowing. So it selects by size and allow the passage of smaller molecules. That is, generally, it is selectively permeable. Cell membrane is a fluid lipid bilayer embedded with proteins. This means, for example, this is a phospholipid, then phospholipid. One layer, the other layer. Consider just like the membrane of a cell, the continuous phospholipids here. So the proteins are embedded in the phospholipid. This, the red is the protein, so it is embedded in the phospholipid bilayer in the membrane. So the major components of cell membranes are the lipid bilayer and proteins. All plasma membranes are made up of lipids and proteins plus small amounts of carbohydrates. The major components are phospholipids and proteins, but also there are small amounts of carbohydrates. Phospholipids are most abundant with lesser amount of cholesterol. Like lesser amount of carbohydrates are there, small amounts are there. There are also cholesterol in small amounts. Cell membrane means plasma membrane. Phospholipids have a polar charged head having a negatively charged phosphate group and two non-polar, that is electrically neutral fatty acid tails. When we say polar, just having positively charged or negatively charged. So the head is polar with negatively charged phosphate. Okay? The phosphate has 3 negative charge. This is a fatty acid head. This is the phospholipid structure. The head part is polar because it contains phosphate, and the tail part is non-polar. The polar end is hydrophilic, that is water-loving, because it can interact with water molecule, which is also polar. The non-polar end is hydrophobic or water-fearing. Phobia means, or phobic means hating or just keeping away from something. Therefore, hydrophobic tails. The tails are hydrophobic or water-fearing and will not mix with water. Such two-sided molecule self-assemble into a lipid bilayer. Okay? Such two-sided molecule means having the head part and this the tail part. Self-assemble into a lipid bilayer. Okay? Having two layers. We will see this layer clearly. A double layer. Okay, bilayer means double layer or two layer. Bi means two, double means two. So double layer of lipid molecules when in contact with water. When it forms, this layer is when it contacts with water. The head part is hydrophilic and the tail part is hydrophobic. Because of this property, it makes double layer. The hydrophobic tail bury themselves. They hide themselves in the center, away from the water. They hide themselves from water. while the hydrophilic heads line up on both sides in contact with water. Both sides means to the outer surface of the cell and inner surface of the cell. Just let's say this is a cell and this is the outer part and this one is the inner part. Okay, so both sides means to the outer side and to the inner side. This is ECF, extracellular fluid. This one is ICF, intracellular fluid. The fluid which found outside of the cell we call extracellular fluid, and the fluids which found inside the cell intracellular fluid. The water surface of the layer is exposed to extracellular fluid, whereas the inner layer is in contact with the intracellular fluid. For example, let's say this is a cell. This is a cell membrane, the outer membrane, which is made up of what? The two components, phospholipid bilayer and proteins. Therefore, let's say this is a phospholipid. The head part is to the outside of the cell, extracellular. This head, this head, this head. By heating the water or the fluids found outside of the cell, it forms what? This layer. The head to the outside, the tail to the inside. And there are also intracellular fluid in the inside of the cell. By heating these intracellular fluids, most of the intracellular fluid is water. So by heating the water, again, the head part is to the water and the tail part is away from the water from inside of the cell. Then it makes what? The first one is this layer, the red one, the phospholipid one layer, and this the green one is the other layer. Okay, the other layer. Just this is I want to show you by extending this membrane. Just this is the membrane. Okay, this membrane is like this. So this is how it makes the bilayer. The lipid is fluid in nature. Okay, fluid means just liquid or liquidy thing. Fluid in nature with consistency like liquid cooking oil. You know, cooking oil in your kitchen. So like that, it is fluid. Cholesterol provides to the fluidity. The cholesterol is in a small amount. It is also the component of the cell membrane. So it provides to the fluidity or to be fluid, as well as the stability, for the stability of the membrane. Cholesterol lies in between the phosphate molecules. Membrane is made up of phospholipid bilayer. So during the formation of this layer, the cholesterol found between the head part of the phospholipid, between the two phospholipids, there is a cholesterol, which prevents the fatty acid chain from packing together and crystallizing. That could decrease fluidity of the membrane. The membrane should be what? Fluid. If there is no cholesterol between this head part, okay, then the tail will come together and be crystallized or solid. Then there will no fluidity. Therefore, the cholesterol contributes for the fluidity of the membrane by this role, by founding in between the head of the two phospholipids of the membrane. Cholesterol also exerts a regulatory role on some of the membrane proteins. Cholesterol is not all bad, okay? Just sometimes it is said that do not consume cholesterol or the cholesterol containing oil. Okay, for example, in advertising oils, they write cholesterol free. That is not mean cholesterol is not totally important for our cell. It is important, but up to some amount. For fluidity of the membrane, it gives flexibility to the cell to change its shape. Also, cholesterol gives flexibility for the membrane to change its shape. So, through the membrane, the movement of molecules from the outside to the inside is important. In that case, the membrane should be what? Flexible. This flexibility comes from having the cholesterol. Transport processes are also dependent on the fluidity of the lipid bilayer. The transport is dependent on not only the fluidity of the cholesterol, also the fluidity of what? The lipid bilayer. Phospholipid bilayer. What are the importance of phospholipid bilayer on the membrane, the cell membrane? Just we are looking on the organelle cell membrane. So one is phospholipid bilayer forms the basic structure of the membrane as a phospholipid bilayer or phospholipid. It is a barrier to passage of water-soluble substances between the intracellular fluid and extracellular fluid, like a wall between two rooms. Between two rooms, there is a wall through which you can't pass. Is that not? Like that, it is a barrier. Phospholipid bilayer will be the barrier like a wall. It is responsible for the fluidity of the membrane. It is important as fluidity maker of the membrane. These are the importance of lipid in the cellular membranes. The membrane proteins. One of the major components of the membrane is proteins. So what are the membrane proteins? They are either attached to or inserted within the lipid bilayer. When we say inserted, just this is a phospholipid layer. Just one layer is here. Consider the whole cellular structure. The inner layer is here. So two layers are here. So proteins are inserted in the phospholipid bilayer, one, and also it can be attached to the lipid bilayer. Here, it is attaching on the surface of the lipid bilayer. It can exist in this way. Some extending through the entire membrane thickness, just like this, extending from one layer up to the other, from here to here. They have polar region at both ends, joined by nonpolar central portion. Here, the protein ends are polar. Other proteins are on either the outside of inner surface to the inner surface of the phospholipid layer, anchored by interactions with proteins that spans the membrane or by attachment to the lipid bilayer. On account of membrane fluidity, many proteins float freely. Fluidity means free movement, or it makes what? Things free. So for this, proteins also found freely. Then membrane proteins, there are variety of different proteins with the plasma membrane. Okay, on the cell membrane or plasma membrane, there are different kinds of proteins which gives different kind of functions. Here are some of the functions. For example, as channels. When you say channel, it is open always. Okay, it is selectively attract or repel particular ions. If the important molecules come, it will pass through always to the cell, or some unimportant molecules come out through the cell always through this channel. The other role of membrane proteins are as carrier molecules that transport specific molecules that cannot cross on their own. Okay, the molecules which cannot pass by their own will be helped by carrier proteins. Carrier means just carry, carrying something. Therefore, proteins will carry those molecules which cannot pass by themselves the membrane, the cellular membrane. So the proteins as carrier molecules. They carry specific molecules. They do not carry everything from the surroundings. They carry specific molecules. Okay? Again, the cellular function is just selectively, we said. Therefore, everything is just specific. Many proteins on the outer surface serve as receptor sites. Recepting means what? Accepting as receptor sites that recognize and bind with specific molecules. Again, here is what? Specific molecules. The function is what? Specific. Here is specific. Here is also selectively. Okay, it is specific. Another group as membrane-bound enzymes. Okay, generally, most of the enzymes, almost all enzymes are what? Proteins. Therefore, the membrane proteins can also used as what? Enzymes. For example, acetylcholine esterase. This ASE indicates what? This is an enzyme. Okay, so acetylcholine esterase is an enzyme which found on the cellular membrane. Some are arranged as filaments. Okay, filaments are, as we have seen, one of the components of cytoskeleton. And we said cytoskeletons are what? Proteins. Therefore, as filaments. Protein as filaments. For example, elements of the cytoskeleton. Others as cell adhesion molecules, abbreviated as CAMs. C-A-M-S. Cell adhesion molecules. Adhesion means sticking or anchoring. Therefore, proteins also used as cell adhesion molecules, which protrude from the membrane surface that grip each other. Grip means catch. Okay, grip each other and grip the connective tissue fibers between cells. We will see this in the next slides. Some proteins recognize self and cell-to-cell interactions. For cellular communication, some proteins are important. Therefore, these are some of the membrane proteins function. This is a plasma membrane or cell membrane. Here you can see the phospholipid bilayer. This is one layer. Here is head, then head, tail, head, tail, head, tail, head, tail. It forms what? A continuous layer, one layer. This is the outer surface, the external part of the cell. Therefore, here you should think that there are extracellular fluids. Outside the cell, there are fluids. By heating the fluids found in this area, it forms what? The head is to the water or the fluid part, and this tail is what? Hiding itself or burying itself to away from the fluid around here. Again, this is the inner part of the cell, and there are intracellular fluids here. By heating or to avoid itself from the water, this tail is forming a continuous chain. Okay, this one is the head part, which is to the intracellular fluid. Therefore, this is one layer. This is the other layer. Therefore, how many layers are there? 2 layers. That is why it is called phospholipid bilayer. And these are proteins. Okay, these are proteins. These are proteins. These are proteins. 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 Okay. This is a cell membrane structure. Here the tree-like structure is what? The carbohydrates. The carbohydrates found only to the external part of the cell, to the outside part of the cell. Okay? There is no carbohydrate to the inner part of the cell. This is just the phospholipid structure. There are 2 parts, main parts, the head part and the tail part. This is the head, which is hydrophilic. Hydro means water, philic means love, so water loving part because it is polar. This is the tail, hydrophobic. So this is just I have shown you previously that the cell membrane, when we extend it, here there is a carbohydrate. If it is attached to the protein, this is the protein, the membrane protein. If the carbohydrates are attached to the protein, we call glycoprotein. Glyco means carbohydrate, here is carbohydrate. This is a polysaccharide, okay, attached to the protein on the membrane, the cell membrane. And here, these are the membrane proteins. Here is receptor molecule receiving some molecule, and this molecule will pass through this protein to inside of the cell. And here it is acting as enzyme. This molecule is breaking down into smaller components. What is the function of enzymes? Just as a catalyst, converting reactants into products. So this molecule is different from this one because of enzymes converting from one form to another. This is one role of the proteins on the membrane. So this is one function. This is as a channel, you can see, as a highway. As a highway, it is passing the molecules which can pass through this protein as a channel. Then gated channel. Gate means what? You know, gate of a compound a campus or a university, there are what guards which do not allow an authorized person to pass through. Like that, these proteins are just as a guards can allow only the authorized molecules. Okay, like guards. This here is a cell identity marker. Here, when we say marker in cells, just consider carbohydrates. Okay, carbohydrate. Here is the carbohydrate as a marker that it is important for recognition of cells. One cell can recognize with another by having these carbohydrates. And here is a cell adhesion molecule or CAM. Here you can see this is one membrane. Okay, this is one cell's membrane. Here is you can see the other cell's membrane. So this protein is extending from this cell membrane up to this cell membrane. So it is making these two membranes to be attached each other. Okay, as CAM. Okay, cell adhesion molecule. These are the pictorial representation of membrane proteins function. Membrane carbohydrates. Carbohydrates found only to the outer surface of the plasma membrane, only to the outer surface. It is found in a small amount. Short chain carbohydrates are bound primarily to membrane proteins. We call these glycoproteins. Glyco means carbohydrate, CHO. Carbohydrate plus proteins, glycoproteins, and to a lesser extent to lipids for glycolipids. Attachment of carbohydrate is mainly on proteins, but in lesser amount to lipids. Okay, carbohydrates plus lipids, glycolipids. Carbohydrates serve as self-identity marker. The main function of the carbohydrates on the cellular membrane is as marker. You know, marker, just marking as a marker, enabling cells to identify and interact with each other the following ways. For example, recognition of self and cell-to-cell interactions. Cells recognize each other and form tissues. Complex carbohydrates act as a trademark, you know, trademark. For example, trademark means the mark which is important to identify one company from the other. For example, you can consider the mark of a Nike products, a mark of Puma. So trademark as a marker of a particular cell type for recognition. For example, you know when cells come together and form tissues, the interrelated tissues come together, form organ. When cells come together, they are what? Attaching each other. For this attachment, one cell should know the other. That recognition is because of what? Carbohydrates. Carbohydrates are also important in growth. Cells do not overgrow their own territory. Abnormal surface markers present in tumor cells. Tumor cells means abnormal cells which are growing abnormally, which will cause finally cancer. And abnormality may underline uncontrolled growth. So it controls cell growth. Some CAMs, cell adhesion molecules, have carbohydrate on the outermost tip where they participate in cell adhesion activity. So these are some of the importance of carbohydrates. Functions of biological membranes. The function of biological membranes are related with the components of the membranes. What are the components? Mainly phospholipid bilayers and the proteins, and in small amounts cholesterol and the carbohydrates. The phospholipid bilayer restricts entry and exit of polar molecules and ion. Okay? Polar molecules will not pass through the phospholipid bilayer because the phospholipid bilayer contains what? The nonpolar tails. In this case, lights attract lights. Okay? As it is nonpolar, it do not attract polar. Okay? The other molecules in the membrane have a variety of function. For example, channel protein, as we have seen, and carrier protein. for selective transport of polar molecules. Polar molecules, as they cannot pass through the phospholipid bilayer, they will pass through what? With the help of proteins. Enzymes. The membrane proteins sometimes act as enzymes. We have seen that. Receptor molecules. Proteins as receptor molecules for chemical signaling between cells. Antigens. Antigens are the substances which are not the part of the cell. They are what? They are just foreign for the cell. They are foreign. So these foreign will be identified by the glyco part, the carbohydrate part of the cell membrane. The carbohydrate is important for identifying or recognizing molecules. Therefore, glycoproteins are important as antigens. These act as cell identity markers or name tag. What is name tag? For example, a badge. You know badge? A badge with a person's name. So it used as name tag. There is an enormous number of possible shapes to these side chains. So each type of cell can have its own specific markers, specific carbohydrates. So it will identify specific things or specific foreign molecules. Cells recognize other cells, for example, during development of tissues. As we said, cells come together form tissues. In this case, it is important. Foreign antigens can be recognized and attacked by the immune system. Our body fights against foreign materials or germs. This is after they recognize them things are foreign. So the recognition process will take place by carbohydrates. Glycolipids involved in cell recognition again. Just as there are glyco or carbohydrate part, it is the same function as glycoproteins or as a marker or recognition. They may act as a receptor site for chemical signals. With glycoproteins, they also involved in sticking the correct cell together in tissues. For example, in this case, energy transfer in photosynthesis and respiration. Proteins take part in the energy transfer system that exists in the membranes of chloroplasts. It is photosynthesis and mitochondria if it is respiration. Cholesterol acts like a plague, reducing even further the escape of entry of polar molecules through the membrane. This tail part or phospholipid is not allowing the polar molecules. In addition to this phospholipid, the cholesterol also controls the passage of polar molecules just by plaguing in between the phospholipid heads. These are some of the functions of the cell membrane. The plasma membrane is asymmetrical. Symmetrical means just the two sides are the same. When we add A, it will be asymmetrical, the opposite meaning. The two surfaces are not the same. The inner surface of the membrane and the outer surface of the membrane are not the same because one carbohydrate is only on the outer surface. Okay, as there is no carbohydrate in the inside of the cell, so the outer surface of the cell and the inner surface of the cells are different. The other is different amount of different proteins are on the outer and inner surfaces. So the amount of proteins to the outer surface of the cell and the inner surface of the cells are different. Even the lipid structures of the outer and inner half is not the same. Therefore, because of the difference in contents of carbohydrates, proteins, and lipids, the inner part and the outer part of the cell membrane are different. They are asymmetrical. Just this is the symmetry. This face and this face are the same. This one is asymmetrical. This face is not exactly the same as this one. So these are the things which makes the cell membrane asymmetrical. For example, carbohydrate found only outside. This is a membrane. So this is the outer layer. This one is the inner layer. So this the outer part. this is extracellular part is different from this the inner part. That is this part, this side, and this side is asymmetrical. Cellular anatomy. The control center is nucleus, assembly line, endoplasmic reticulum, workbenches, ribosomes, distribution center, Golgi complex, security gate, cell membrane, like the guards in your campus, cleaning crew, lysosomes. It is important for cleaning the wastes or unwanted material inside the cell. Powerhouse, mitochondria, and structure, cytoskeleton. These are the analogy of the cell membrane, which you can easily understand. Cellular diversity. This refers to number. When you say diversity, just refers to number. Cells are very diverse. That is, many in number. In their size, there are different or many number of cells when we consider size. Many number of shapes and their internal structure. Cell shape, cells can have this kind of shape: flat shaped cells, long shaped cells, spherical shaped cells, rod shaped cells, and rectangular cells. So depending on these features, cells are diversified or many in number. For example, this kind of shape is called squamous. This kind, when you look at this is called squamous kind of shape. Cuboidal, this kind of shape. Cubic, or having four sides equal, so cubic kind of shape. Columnar, column-like. It is long to the column. Okay, columnar shape. This one, this one, this one. And polygonal, having many sides. When you look at this, there are many sides. Okay, each cells are many sided. So poly means many, gonal means side. So polygonal. Stellate, star-like structure or star-like shape. Spheroidal, just like a sphere. A sphere means the globe-like structure. Discoid, disc-like structure like red blood cells. Fusiform or spindle-shaped, just spindle-like shape. And fibrous, fibrous means like thread or fiber, long and extended. These are just to show there are diversified kind of cells by their shapes. When we look at the cell size, generally we know that cells are very small or microscopic. Then there are also large cells. They can be seen by our naked. Naked means without using any magnifying tools like hand lens or microscope. So depending on their size, there are also diversified or many number of cells. So generally cells are diversified. Transport across cell membranes. There are four basic mechanisms in this. So they are diffusion and facilitated diffusion, osmosis, active transport, and bulk transport. Generally, when we consider about transport, just we should consider the membranes, okay, cell membrane, because it is what is through which the particles or the molecules pass through. When we consider or when we say cell membranes, we have to consider the properties of the membrane components, such as the phospholipids and proteins. Therefore, the transportation is related with these components. When you say diffusion, simple diffusion, such as, for example, spraying perfume and dissolving of salt in water, they simply pass from their higher concentration to lower concentration or move from their higher concentration to their lower concentration. When you say facilitated diffusion, there is what? Facilitation by proteins. Okay? Protein molecules help the movement of the particles or the compounds facilitated. And the other is osmosis, just the movement of water across the cell membrane. When you say osmosis, the movement of water from the higher concentration. By nature, water moves from where they are higher in concentration to their lower concentration, that is osmosis. We can classify this diffusion and facilitated diffusion and osmosis into what? Passive force. There is no need of what? Another force for the movement of these molecules, because molecules are always in motion. Okay, there is kinetic energy. So they collide. They collide with the container they found, or they collide with the membrane. Then there is what? Kinetic energy. Kinetic energy refers to what? Motion or movement. Just relate with your physics course. Therefore, molecules collide. Then during collision, they pass from their higher concentration to their lower concentration without help of energy or without requiring energy. That process we call passive transport, or by the passive force. Then active transport, it uses what? ATPs. Just simply when you say active transport, consider there is what? Utilization of ATP. The other kind of transport is bulk transport. Bulk means large sized. So the movement of large sized molecules is through the cellular membrane. So let's look at each of the transportation mechanism. Diffusion is the net movement of molecules down a concentration gradient. That means from the higher concentration to the lower concentration. There is gradient or differences between two parties. Gradient means difference. The difference between the two parties, between the extracellular and the intracellular. There is concentration gradient, therefore they move from their higher to lower concentration. Certain additional factors. It says additional factors. What is the main factor? The main factor in the movement of molecules in the diffusion cases, that is what? The concentration gradient. The difference between the concentration of molecules to the outside and inside of the cell. So the additional are permeability of the membrane. If the membrane is very permeable, then the movement of the molecule through the membrane will be high or increase. Surface area of the membrane, okay, SA. When the surface area of the membrane increases, then the movement increases. Then molecular weight of the substance. The substance which is passing across the membrane. The lighter molecules can diffuse rapidly and easily. Okay, the larger sized molecules diffuse slowly. Is that just a very simple concept? Distance through which diffusion must take place. That the distance is what? The thickness of the cell membrane. If the thickness increases, the diffusion decreases. Okay, so these two are directly proportional and these two are inversely proportional to each other. That means when permeability of the membrane increases, the movement of the substance or the molecules increase. When the surface area increases, the movement of the molecules increase. In this case, when molecular weight of the substance increases, then the movement decreases. And when the distance of the membrane or the thickness of the membrane increases, the movement of the molecules decreases. For example, concentration difference between inside of the cell, this is the outside of the cell. Therefore, here is movement of molecules from its higher concentration. This one is empty, or there is no molecule. Therefore, there are a lot of molecules here. It will cross this membrane and then pass to this side. Here is diffusion. Because of molecules collide with this membrane, then when they collide with this pore, then passes to this. Diffusion of lipid soluble molecules. For example, this is outside environment of the cell. This is the cell membrane. This is the inside environment of the cell. If the molecules are non-polar, then they can pass across this membrane. This is a cholesterol, which can allow the non-polar molecule. So through this, and this part is no problem to pass the non-polar because they are non-polar. So likes attract likes. Therefore, non-polars can pass through this one. So again, this one, the same thing. Facilitated diffusion. Facilitated diffusion uses carrier protein to facilitate the transfer of particular substance across the membrane downhill from higher to lower concentration. You know heel. This is a heel, for example. Okay, a heel. So downhill means from this part to this part without requiring any energy. It can roll down, yes, roll down here. This process is passive and does not require energy because movement occurs naturally down a concentration gradient. Very simple. Osmosis, net diffusion of water. Here, for example, the red one are water molecules, and this one are the sugar molecules. This is the cell membrane. Okay, this is the inside of the cell, and this one is the outside of the cell. So the water will move from this higher concentration to this lower concentration. Only two reds are here, so less amount of water here. Therefore, we expect movement of water from this part to this part. So this indicates that. Active transport is energy consuming transport. Okay, it consumes ATP. The movement of molecules is in one direction only. So there is no equilibrium because with the help of energy, it only passes to one part. The major ions within the cells and their surroundings are sodium ion, potassium ion, and chloride ion. Okay, this passage of these molecules require energy. The cell surface membrane of most cells have sodium pump, which is coupled with potassium. The sodium pump is coupled with potassium pump, which is called sodium-potassium pump at the same time. The pump is carrier protein. When we say pump, the molecule is what? Protein. 2 sodium ions taken into the cell from outside and 3 sodium ions are removed from the cell. Thus, a potential difference is built up across the membrane because they are not equal. 3 are taken out from the cell and 2 are entering into the cell, so there will be a potential difference, with the inner side of the cell being negative because only 2 is entering and 3 positives are taken out from the cell, so more negative inside. This tends to restrict the entry of negatively charged ions because there is one low concentration of positive ions inside, therefore positive ions will enter into the cell. The negative ions favoring diffusion of cations. Okay, the positive ions. Cations are the positive ions. This explains why chloride concentration inside red cell is less than the outside, despite the fact that chloride ions can diffuse in and out by facilitated diffusion. Therefore, here don't forget that 3 sodium come out from the cell to outside, then 2 potassium come into the cell. 3 sodium, 2 potassium. The sodium-potassium pump plays 3 important roles. 1, it establishes sodium and potassium concentration gradients across the plasma membrane of the cells. So maintaining the sodium-potassium concentration is important in cells, so it maintains that. Important is the nerve and muscle to generate electrical signals. It helps regulate cell volume by controlling the concentrations of solutes inside the cell and thus minimizing osmotic effects that would induce swelling or shrinking of the cell. So it is important to regulate cell volume or cell growth. The energy used to run the pump also indirectly serves as the energy source for the transport of glucose and amino acids across the membrane. So energy in the form of ATP. We are looking at an example of active transport, sodium-potassium pump. Therefore, the energy or ATP used in this process also used in transporting other molecules such as glucose and amino acids. Just consider that glucose and amino acids are important to make ATP inside the cell because in the cell, the glycolysis, which takes place in the cytoplasm, and the TCA cycle takes place in the mitochondria, which is within the cell. This is a sodium-potassium pump. This is the outside environment of the cell. It says outside cell. So there are a lot of sodiums. Okay, sodium ion. There are a few amount of potassium. This is the inside cell. There are a lot of potassium. This is a sodium-potassium pump. This is a protein or the pump through which the sodium come out from the cell to outside and the potassium come into the cell. So a lot of sodiums are coming out, then a lot of sodium outside environment, and a lot of potassiums are entering into the cell, therefore a lot of potassium inside. Exocytosis and endocytosis. Endocytosis and exocytosis are active processes involving the bulk transport of materials through membranes, either into the cells, that is endocytosis, or out of the cells, or exocytosis. Endo means into, exo means out of. It is also called vascular transport because it forms what? Vessel. Vessel is just like a container or just like your bag in which you can put something and transport. So it is vascular transport. Process of transport into or out of the cell is a membrane enclosed vessel. Okay, the vessel which has membranes. Endocytosis transport into the cell, whereas exocytosis transport out of the cell. Endocytosis of fluid we call pinocytosis or cell drinking, and endocytosis of large multi-molecular particle or solids we call phagocytosis or cell eating. This is cellular environment, and just exocytosis of waste. Waste is not important in cell, so there will be what? Coming out of this waste as bulk. Okay, and here food particle taken into the cell. So this food will come into the cell membrane, then it will form this vessel. So this part will enter into the cell. This is what we call vascular transport. And this one is also the same. So this one come out from the cell as vessel. Thank you so much. This is all I have for today. If you have any question, please write in the comment section of the video. Please don't forget to subscribe to my channel. Bye bye. Have a nice time.